Okay, so hi everyone, my name is Darby. My project is called Multiscale Rheology of Soft Biological Materials. I've been working with Dr. Jamal Ali and his postdoc student, Dr. Nanjyoti Kakati, and my project focuses on characterizing different materials via their viscoelastic properties. And this method is called rheology, and there's a bunch of different ways to do it. And we wanted to make sure that all these methods had similar results, confirming each other. There we go. So first off, just about each of the methods we use. The first one is called bulk rheology. So this is done using an instrument called the rheometer, and you can see that in figure 2. So you compress the sample between two plates, and the top one spins, and then the relationship between the strain and the resistance of the material gives us insight into the viscoelastic properties. And then in microrheology, you use video processing to analyze the motion of the particles. In passive microbiology, you're just taking video data of the Brownian motion of the particles, which is how particles move naturally in a fluid via Brownian motion, and that's seen in figure 1. And then in active microbiology, you take one particle, you do the same video processing on it, but you use optical tweezers, which is what we have in the lab, to move it around individually, and you can see the difference in the relation between the force and the resistance of the material to give you insight into its viscosity. So some of the methods we used, and I actually got trained on all these methods, which I haven't experienced before, which is super cool. So I got to learn how to do cell culturing, bulk shear rheology, different kinds of microscopy, particle tracking microbiology, which you can analyze using Python and MATLAB and Excel and Origin. So that was great practice as well. And then optical tweezer microbiology as discussed. And then I also got to learn how to use a scanning electron microscope, which is super cool. So in figure 3, again, you can see how that material gets squished with the bulk shear rheometer. And then in figure 4 is how we prepared slides for the microscope. So in those little diamond-shaped holes, that's where we put the sample, and we put a cover glass on top of it. It squished it in there and kept it nice in place for use in the microscope. And then figure 5 shows a real image of the fluorescent tracking particles that we use to analyze it. And then in figure 6, you can see how the optical tweezer applies a force or strain to the particle, and then you measure the resistance from that. So the results we got, we chose some materials for calibration, including glycerol, alginate, and polyethylene glycol. So in figure 7, you can see our values in red and black very closely aligned to the values in previously reported values in green, which means that we were on the right track. And then in figure 8, we looked at the mean square displacement of all the particles, and you can see that the mean square displacement increased as viscosity decreased, which makes sense. And you can see the chemical structures getting more and more complex as well. And then finally, we compared multiple particle tracking, rheometry, and optical tweezer microbiology in figure 9. And you can see that all of the plots are nearly continuous, which means all of our results align with each other. And then in conclusion, we did calibration with some of the simpler materials, and we got to start using doing analysis on Matrigel, which is a very difficult to work with biofilm that's used a lot in cancer research. It's derived from mouse cells, and it's really hard to work with because it's liquid at around fridge temperature, and then solidifies at room temperature. And then in that last figure there, you can see that all the different techniques fell within a standard area of each other. So thank you everyone for a great summer. I had a great time, and this was a fantastic project.